All right, next we're going to be looking at the histology of the nervous tissue. There's two principal cell types: neurons, kind of the major cell for nervous tissue, and neuroglial cells. Whereas neurons are very excitable, they have the ability to transmit electrical signals and cause the release of neurotransmitters, neuroglial cells were once thought just to be glue, kind of a supportive or structural component to the nervous tissue. But now we know that there are 6 types, 4 in the central nervous system and 2 in the peripheral nervous system, and they do much more than just hold everything together. They actually have their very own important functions. So let's look at the quick list at the left of the different neuroglial cells. You have astrocytes and microglial cells, ependymal cells, and oligodendrocytes. All of those are found in the central nervous system. But you also have satellite cells and Schwann cells, and those are found in the peripheral nervous system. So here we kind of have this glue of the nervous system slide where we have a phrase to just help us remember all of these 6 cells. So the phrase is, glial cells hold the central nervous system and the peripheral nervous system together at the seams, with the S for satellite cells, E for ependymal cells, A for astrocytes, M for microglial cells, and because Schwann cells and oligodendrocytes have the same function, we put them together for the second S. So let's talk about each one. So first we have astrocytes. They're very abundant. They're the most abundant glial cells. They're versatile. They kind of do it all. They're the mini supermen or men of the nervous system. They're very highly branched glial cells, and they're able to cling to neurons or the synaptic endings. They even cling to capillaries inside the brain. So their functions you can see here are many. They support and brace neurons and capillaries. They play exchange roles between those two structures. They even guide the migration of young neurons and control the chemical environment around neurons, including those young ones. They also respond to nerve impulses, neurotransmitters, and by doing so, they influence neuronal functioning. And that allows them to participate in information processing in the brain. So here's what they look like. We can see a central cell body of the astrocyte with a nucleus, and it has many different outcroppings or branches, and some of those branches hold onto capillaries or neurons. And this dual function here and how they're both supportive but structural is going to help them participate in neuronal functioning, especially when it comes to the transfer of information or even the transfer of ions. We also have microglial cells, and the big thing to know about microglial cells is they are phagocytic. They can transform to phagocytose microorganisms and neuronal debris. They actually go toward injured neurons in order to do so. They're a little bit smaller than our astrocytes, but they do have a lot of processes like our astrocytes. But their soma or cell body is much smaller and ovoid in shape. So we can kind of think of a microglial cell as a neuron nurse with a thorny side, whereas astrocytes are kind of the do-it-all and take care of everything. Microglial cells are going to take care of things that are breaking down. So here's our microglial cell. You can see it's just monitoring nearby neurons, and if they were to break down or send out certain signals, it would migrate closer to them and start to break them down via phagocytosis. Next we have ependymal cells, and this may kind of look like the term epithelial cells, and ependymal cells do have a similar structure. They range in shape from squamous to columnar. Where have we heard that before? With epithelial tissue. They might have cilia, like some epithelial tissue, and those cilia beat to circulate cerebrospinal fluid in cavities of the brain and spinal cord. And where we can find them are in the brain and spinal cord. But they do have that epithelial sheet shape. These cells are going to line up next to each other, and they're going to be the inside lining of central cavities for the brain, central cavities for the spinal column, and they're going to form a permeable barrier between these spaces with cerebrospinal fluid or CSF and the tissue that is bathing other cells. Now, ependymal cells definitely should remind you of epithelial cells because they do both like to form permeable barriers. They contain extensions like cilia, and their functions, like moving fluid back and forth using those cilia, or being an important sheet and protective in some way, or being secretory or absorptive. Those are things that are both seen in epithelial cells and ependymal cells. So here's our ependymal cells. We can see a sheet of cells, roughly columnar in shape here, embedded in brain or spinal cord tissue with cilia on their apical surface, and that's going to be near a fluid-filled cavity, and that cavity usually contains CSF, which is cerebrospinal fluid. All right, next we're going to look at our oligodendrocytes. Oligodendrocytes are branch cells, but they're not as branched as astrocytes or microglial cells. Instead, they just have a couple of branches. And when these branches come into contact with the axons or long processes of neurons, they start to wrap around it and form what we call a myelin sheath. A myelin sheath is kind of a lipoprotein, kind of like a fat and protein together, that is going to be very protective of axons, but also conductive. It's going to help speed up the action potentials or electrical impulses that carry down these axons. Now, oligodendrocytes make myelin sheaths in the central nervous system. Actually, all the cells we've been looking at, astrocytes, ependymal cells, microglial cells, and now oligodendrocytes, they are found in the central nervous system. But Schwann cells, which are really similar to oligodendrocytes, are found in the peripheral nervous system. But they also make myelin sheaths. They do so in a little bit of a different way. They create a jelly roll kind of structure around the axon, and you need multiple Schwann cells to get most of the axon for a peripheral nervous system neuron coded. But Schwann cells and oligodendrocytes do share that similar function of creating myelin sheath. And then we also have satellite cells, which are also in the peripheral nervous system, and they're more kind of similar to astrocytes of the central nervous system, but otherwise very similar to cells we've already talked about. So satellite cells kind of do it all and are supportive and protective for our peripheral nervous system neurons, and are oftentimes found around the neuron cell bodies in the peripheral nervous system. They kind of go around it like a satellite goes around Earth, hence their name. So here's an image that shows both. We have the purple satellite cells going around the cell body of this neuron. And you can see the Schwann cells going around the axon, forming the myelin sheath. They kind of look like small little marshmallows, but each one of the marshmallows would be a Schwann cell. All right, next, we're going to look at neurons, so make sure to come back for that video as well.